All righty, Keith. Thanks so much for joining us today. It is so good to see you again. Why don't we start with a brief background on who you are and how we ended up chatting and what you're doing today?
Right. So I'm Keith Robison. I'm currently in a product role uh, at Ginkgo Bioworks in their automation group. So I've been at Ginkgo since 2019. I started my career, I did my undergraduate at the University of Delaware and then came up to Harvard for grad school. And then that bunch of people I met at Harvard pulled me into Millennium Pharmaceuticals, where I spent 10 years from 1996 to 2006. Then it was 2 years at Codon Devices, which was a synthetic biology startup that didn't make it. We ran out of money in the middle of the big financial crisis. And so then I went to Infinity Pharmaceuticals, which was another drug discovery firm. And then I thought I was going to be just stay there. And I got lured away to by a former colleague to a startup called Warp Drive Bio, where I was one of the first five scientists. We didn't have labs when we started. It was quite an experience. And then Warp ended up running its course and being acquired by a company called Revolution Medicines, which then sold my kind of part of Warp, what I had helped build, to Ginkgo, and that's where I ended up at Ginkgo. And along the way, I've written a blog for almost, it'll be 19 years at the end of this month. It might be 19 years about today. I need to go.
Congrats. What a great way to celebrate.
Yeah, yeah. No, this is fun, and I've been an advisor to a company that started and ended, but it was still that's an interesting experience, and yeah, it's been a lot of fun. It's been ups and downs, but it's been an interesting career, and it's not over yet, but
Of
it's
course.
it has been interesting.
Uh, you've been in the biotech world for so long. A goal with this podcast is to kind of make biotech a bit more legible to the folks who don't belong to the bubbles of South SF and Cambridge. These are smart people that understand technology but do not understand biotech. I think it would
Sure.
be really helpful to get a sense of how you think biotech has changed in the years when you began working versus what it is today. And what have been the most surprising changes that you've witnessed in your career?
Yeah, I mean, it has been huge. And of course, I mean, Millennium, so those 10 years, we're almost evenly split. By the first 5 years, we are trying to anticipate the initial draft of the human genome. And to some degree, it was a land grab, or at least be able to make sure that we could have freedom to operate. And so I used, and there's a couple tales I can tell there, but yeah, so we were trying to mine the human genome faster than everyone else. And then the first draft of the human genome gets announced, and there's all sorts of surprises there. Genome smaller than we thought. A lot of people had, or not smaller in terms of total base pairs, but there had been a lot of, in some degree, self delusion of how many genes are there going to be. And so this
Mm,
number, and I actually had a colleague who did this analysis. Tom Barnes, he's off on his like third or fourth company now. Genius guy. And he did this analysis of where, tried to figure out where did this number of like 50,000 genes that was in the textbooks come from. He couldn't, as far as anyone can tell, it was an off the cuff remark by Walter Gilbert, Nobel laureate for developing sequencing, and one of my advisors. It was just an off the cuff remark. Someone asked him how many genes he's just like, well, I don't know, maybe 50,000. Got into the textbooks. And then there were these companies selling databases of genes. And so it was in their interest to keep, and they kept doing it. They're saying, oh no, there's 75,000. Oh no, our data says there's 100,000. And of course, that made their data more valuable. And so Tom does this analysis and comes up with this argument that it's only like 20 to 25,000 genes. And I'm like, Tom, I can't figure out what's wrong with your reasoning, but you've got to be wrong. Of course, I was wrong. I like to kind of keep track of times in my life I've been really wrong because I think it's important. And boy, this is one of the bigger ones because Tom was quite right. Comes out, the genome is much smaller. But then it was like, okay, we know, and of course, to some degree, the human genome wasn't truly finished till a couple years ago. The original genome goal was to get the bulk of the genome, and ignore the really complicated stuff because it wasn't technically possible. And there were mistakes and all that stuff. But it was still this watershed moment. And then it's like, okay, we have the genome. What do we do with it? So to some degree, one thread going through a lot of things is over, since 2001 or whenever that announcement was, 2000, 2001, is how do we exploit the human genome? Of course, that's still an evolving story.
Mm.
And then the other big one that's intersected me a lot is subject of the blog is the genomics technology just keeps getting better and better. You can, in particular, you can collect more data with less work and importantly less cost, so you can do much bigger studies. So it was this huge event during my at the very end of my thesis. In fact, I ended up rewriting my thesis because of this, that or part of it. But the first 3 bacterial genomes were done. And like we participated with E. coli, and that was like working on E. coli was this multi-year project to get the first E. coli genome, to get one of the common lab strains done.
Mm.
And it was a big deal when Craig Venter's group, after knocking off Haemophilus influenzae, did mycoplasma, one of the mycoplasmas, and they did it in a month of data collection. A month. And that was amazing. Well, now, somewhere like Ginkgo, we routinely sequence thousands of bacteria a day. You can knock off a bacterial genome on an Oxford nanopore device in, if you really want to, like focus on one and, a few other things all lined up, but one person can do it in like a matter of an hour.
Yeah. Before we, so I think the arc here is that sequencing has gotten cheaper, faster, easier. There's more genomics data. Before we get into the depths of it, what is sequencing? I know what it is. I think I know what it is. Okay. But I think for those who are still new to the space, let me give you my definition of it, and maybe you'll have some corrections. Sequencing is just the ability to understand the order of A, T, C's, and G's within our DNA, and that's applied at the DNA level. And you have multiple layers of complexity in biology. You have the DNA level, you have the RNA level, you have the amino acids, you have the protein level, and each level requires a different sort of sequencing because they interact in different complex manners. So what you just referred to with bacteria sequencing was specifically around sequencing the DNA of these bacteria. Does that sound right?
Yeah, so and that's a good foundation. I mean, it keeps building from that. So like one direction, of course, we can go is with some of the sequencing technologies. So you have A, C, G, and T, but now you can also read out these modifications that are epigenetic modifications.
What is epigenetic? What are epigenetic?
Right. So what's epigenetics? So your genetics, and people argue over the definition. So I will try to define it.
Yeah.
So you have your genetics, like you have your genome, that's DNA. And this also gets to a fundamental question. There was a class at Delaware that I really wanted to take. And I talked to the professor, and he's like, well, if I don't get enough students, then I'm not going to run it. And so I started going around to other students in my program, because it was like one of these, do so many of this group to sell the class. Like I can turn into sales mode if I need to. And like it was a developmental biology class. And I said, here's the fundamental question in biology. That's absolutely fascinating. But if you're taught, if you've had like a high school or junior high biology or anything like that, you're taught that a cell exists and then it divides into 2 cells, which are identical.
Yes,
mitosis. They divide into 2 cells.
I remember this.
Right. So you and I are obviously composed entirely of identical cells, right? Well, of course not. Of course we aren't. So that's where things like epigenetics come in, and that's where some of the arguments come around. But basically, there are important. You have that order of A, T, C, and G, but there are other things that go on. So like in our cells, for example, there are marks that can be, there's 2 major chemical modifications to C, and they don't change how it pairs. That's the important part. That's what makes it epigenetics, but it is stable to a large degree, and that is one way things are modified. Also, the DNA, there's these, I don't know these numbers offhand. If you took the DNA in a human cell, stretched it out, it would be like meters long, but our cells are not. So it needs to be tightly compacted. And so it's wound around these proteins called histones, and there's levels of that. But an important part of that is how tightly is it wound? So you have open chromatin, and that's basically accessible to be expressed. And you have closed chromatin, which is not accessible. And so that's another layer of epigenetics. And then, you can look at the histones themselves will have marks on them, which are part of this process, and so forth. So you have layers and layers. If you look at why everything's kind of adjustable.
Understand epigenetics today. We started this conversation talking about sequencing, and you specifically brought up epigenetics. What are some ways in which we understand epigenetics today, or sequencing?
Yeah, let's see. What are some of them? Well, okay, so like one really fascinating and serious bit of it is, so another thing, one way I always look at genetics is, and especially this is the thing that everyone should be very humble about whatever they learned in like high school genetics or freshman genetics or senior year genetics or PhD genetics,
Oh
because it's
wow.
always more complicated. So just
Yeah.
always assume that if someone who has more experience contradicts what you were taught, well, there's probably an exception. So like one fascinating and serious bit of epigenetics is that there are, most genetic conditions you're taught, it doesn't matter which parent you get them from. There's some that are kind of preferentially inherited from one side, that's the way chromosomes are. So X-linked disorders tend to get from your mother, but otherwise it doesn't matter which parent you get them from. There's a small subset of genetic disorders that violate this, and I'm going to blank on the names of any. Prader-Willi is one of them, and it has to do with epigenetics. And it's basically due to epigenetics, one copy is silenced. So if you get from one parent, and that's distinctive from that parent.
Yeah.
And so if that happens and you get a bad copy from the other parent, then you get the disorder.
Well, I was more asking about how do we sequence or.
Oh, I know. Let me just throw out the one really visual that's cool. So, of course,
Yeah.
all mammals have the same sort of epigenetic imprinting. So if you've ever calico and tortoiseshell cats, this is the classic example. That color pattern is due to epigenetics.
Okay. That's
It's due to
cool.
exon activation, which is an epigenetic phenomenon that in females, mammals, you have two X's, but one gets shut down, so you don't get a double dose, because otherwise there'd be a problem, because males only have one. So you need to half as much. So chromosomes are randomly inactivated, and in those cats, there are color genes, they get inactivated, and so you get these patterns. So that's cool. So how do you read epigenetics? Well, there's a bunch of ways, and we could go really deep in that, but one of them is these methylation marks on the DNA, methyl C. Some sequencing technologies can read them. So like PacBio and Oxford Nanopore, they change the signal and you can read that out.
Yeah.
For other technologies, there are ways to basically treat the DNA in advance, so you get these patterns of, if this was methylated, if it was a methylated C, we'll read it out as T, but in the opposite strand, we'll still read G, so you can kind of infer it from there.
When you say methylation groups, these are just compounds, these are CH4 compounds.
It's a CH4,
Yeah.
and it's added to the nucleotide, but in such a way that it doesn't change the base pairing.
But it changes the way that it's read. So
there are proteins that go and recognize that methyl is on there, and then there's another one called hydroxymethyl, where you have a hydroxyl group on the methyl. That's red. Again, once there's differences, you can also, like, just an aside, there's this cool set of tricks that are sometimes called molecular stenciling, go under other names, where we artificially add modifications, so that you can also, in bacteria, you can also modify A. That doesn't happen in eukaryote. And so what you can do is you can extract chromosomes very gently, so you preserve this higher order structure, treat them with an enzyme that will methylate the A's, but that enzyme can't get into the closed regions. So only open chromatin will get. And now with PacBio or Oxford Nanopore, you can read those out. And so you can see, and this is cool, like you get the bases, and you also go, oh, that's a methyl C. And look, because we treated it this way, that position, all these A's are methylated, therefore this is open chromatin. All these A's are not methylated, therefore that's closed chromatin,
right,
and so forth.
Right. Yeah, that's fascinating. But I think we need, when I first started learning about all of these sequencing technologies and how do we read the code of life, all of it seems super interesting and super fascinating, but it wasn't super intuitive to me as to why this is important. So I think we ought to talk a bit about the drug development process and where these sequencing and all these omics technologies fit into the drug development process, and then we can talk more about sequencing specifically. But you started your career in this world. What did the process used to look like and change now?
Right, and this is something that's changed because of advances in other areas. So kind of millennium, what I was the computational head of was this idea that if we, at that time in particular, if you looked at in 1990, say the late 90s, here's the world of pharmaceuticals. That most pharmaceuticals are small molecule drugs, because for most indications, if you want,
what is an indication? Sorry,
indication. So for most diseases, like an indication is a specific like disease. Like I'm going to treat asthma, I'm going to treat eczema, I'm going to treat cancer, and so forth. So for most of the, but cancer would be pancreatic cancer or non-small cell lung cancer. You try to subdivide that to the greatest specificities, because that can make a huge difference.
Right.
So, and a lot of it, if you look at the market, then almost all drugs on the market were what we call small molecules.
What does
So
that?
that means
Yeah.
organic molecules under, mostly under 1,000 kilodaltons. So 1,000 times the weight of, sorry, 1,000 daltons, 1,000 times the weight of hydrogen.
Okay.
And roughly half of those came from natural spaces where, what did nature come up with? So you think of almost all your antibiotics. Apomycin's a drug people love to talk about these days. That's a
Yeah.
natural product.
Along with
A lot of
longevity
those,
drug, right?
right? But then there's also a lot of drugs that that were developed by human in, cleverness. And so one of the things about with most disease areas and most, you come up with a product idea that I want a drug that's going to treat this disease. That for most of those, it needed to be orally bioavailable. You need it to be able to deliver it as a pill.
Yeah.
It's just commercially not feasible. Like if you have an asthma drug and you're saying, "Oh yeah, you're gonna have to come to your doctor's office once a day to get an injection," yeah, that wasn't going anywhere. So like oncology, for example, was an exception. Also in that era, but you were starting to see so one of the interesting signs that was starting to happen. There were starting to be some of the first antibody and protein therapeutics. There have been some protein therapeutics for like rare diseases. So like Genzyme built a whole company around enzyme replacement therapy, where patients with a rare disease are lacking an enzyme, Gaucher's disease, and you give them that enzyme. And if you can make it, again, this is a really serious disorder, and you make it so that the infusions are rare enough, with some proteins you can tune it, so it's maybe you have to go to your doctor's office once a month. And if the alternative is really serious disease, that worked. But one of the things you started to see was auto injection, and the first auto injectors came out.
What is an auto injector?
So this is so if you know anyone on like GLP-1s, like
Okay, got it.
Ozempic or so forth,
Yeah.
there are basically these pens
Yeah.
that allow you to inject yourself. I mean, even before that, of course, the diabetics had long been injecting themselves with insulin, but you're starting to see that. And of course, what that's opened up, and with my son, I'm watching sports a lot of times, and you see a lot of ads for a lot of antibodies.
Yeah.
And that's because, and they're not for cancer, they're injectables, but now you see injectables for a lot of autoimmune disorders because these like auto injection type strategies work. So again, if for something like eczema, you probably aren't going to want to even go into the doctor's office once a month, but even severe eczema. But if you can inject at home and people are willing to do that, that's been destigmatized, et cetera, et cetera. So, but anyway, so back in the millennium days, the idea was if you look at small molecule drugs, they only affect certain types of proteins. So you could go through all the known human proteins, and as we discovered more, and this is what I developed software for, and basically classify them into druggable and undruggable. And you may have heard this term a lot.
So we're right now squarely in the drug discovery phase.
Yeah, we're in the drug discovery phase.
Trying to figure out which molecules actually make sense and actually.
Right. So you'd have this problem, and this is like Millennium started out using human genetics to discover drug targets. Because the idea being, if a gene, when broken, causes a disease, or as the other, which really came out later. Why am I blanking the name? So a big area for cholesterol reduction came from a study of African American families, and they realized these families had really great cholesterol numbers no matter what they ate, and it turned out they're knocked out for blanking on it. They're knocked out for a gene. That kind of genetics would tell you a lot about things. But the problem, like Millennium found when they did these genetic studies, is a lot of times you'd end up with a gene, you'd be like, this is an undruggable target. What do we do with it?
When you say undruggable target, you mean we can't alter this gene?
Well, at that time, the gene looked like that type of protein, no one had ever successfully developed a small molecule drug against it. If you looked at all the small molecule drugs, they typically went to a handful of different types. So like certain receptors on cell membranes, ion channels, certain enzymes, and not even all enzymes, just certain enzymes, and a handful of others. But for example, just some disorders are due to bad collagen, the protein that is in connective tissue. No drug had ever been made against collagen. So what's happened since then is people have been clever. And again, like one of these other stories of me being really, really wrong is in the early 2000s, and I'm in the midst of all this. And there's this announcement that the Cystic Fibrosis Foundation is going to work with a small company called Aurora Discovery. If I remember right, that was the name. It was soon acquired by Vertex. And
Mm.
they're going to develop a drug against cystic fibrosis, which is a case of a broken gene. And no one had ever had a drug that repaired a broken protein. And so I was like, no, that's never going to work. Well, of course, Vertex has been insanely successful with that. It's been amazing for cystic fibrosis patients. It's changed, they've gone from having not expecting to live beyond about 30 or mid 30s to they'll probably have full lives. CF has this strong infertility effects in both males and females, and the drug basically changes that instantly. And so it's amazing because Aurora and Vertex scientists were not so convinced that you could not drug this, and they figured out a way to drug it. And it's basically a drug that enables these proteins, it stabilizes them enough that they don't get destroyed. Another one, which Warp Drive I worked at, was an early company or what we call molecular glues. And so you find molecules that have two halves. One half binds one protein, one half binds another, and you bring those two together. And if you bring those two together, for example, you might block one of them from functioning, because someone at Warp Drive compared this to, the person who parks diagonally across 3 parking spots and blocks the the EV charger. That's a really obnoxious behavior, but that's what we're trying to do. We're basically trying to bring that car next to the EV charger so that no one can use it. I mean, in this case, it would be block a cancer triggering protein. In other cases, you might drag a protein over to a protein that'll cause its destruction. And other times you might drag a protein over to a protein that will enhance its lifespan. But just to finish the thought, so these things, PROTACs, these molecular glues and so forth, are one of many things that have just expanded the definition of what we can drug. So now the idea is just get the biology right. If you can figure out something with the biology, then with RNAs or DNA, gene therapy, PROTACs, antibodies, etc., get the biology right, and everything's druggable now, or nearly everything. I think modern pharma no longer thinks in terms of druggable, undruggable, because the idea is the only thing limiting what we can do therapeutically is biology, our understanding / ignorance of it.
That is a great point. Something I've thought a little bit about. In the one thing I've realized in learning about all of this is how random and unpredictable so much of this is. And my understanding now is that drug discovery, even drug development, has been kind of just throwing the kitchen sink at it and seeing what works. Is that the right characterization? Where does sequencing tooling fit into all of it?
Yeah, so I guess some of it. I mean, at different stages. So there are things. So there's one class of drugs called ACE inhibitors, and they were actually, the biology was figured out.
Yeah.
And then they started taking the protein that ACE was targeting and very rationally designing it. Vertex was built on the idea of so-called rational drug design, which is very different than throwing the kitchen sink.
Yeah.
But there are some approaches that's not a terrible description. There's also a lot of drugs that were discovered that were intended for one thing and discovered for another.
Minoxidil.
Yeah, minoxidil is a famous example. Viagra is a famous example. But the whole family of antidepressants started originally as an anti-tubercular drug, and volunteers on the trial reported feeling euphoric.
Oh, wow.
And that led to if I remember right, it's the tricyclics, which then helped. The other thing about every time you get a drug, now you try to figure out the biology of the drug. That reveals all this biology, which you can then use again. So tricyclics turned out to interfere with both dopamine and serotonin, or alter that processing in the brain. And that led to the SSRIs, which is like Prozac. So there's a lot of chance and luck. There's also another story I love to tell, and it mattered a lot to me because at the time I knew someone who was going on a heart transplant. But I happened, it was one of these flipping through the channels kind of things. I stumbled on this documentary, and they're talking about how at Sandoz, which was one of the precursors to Novartis, the scientists had this compound they'd gotten out of a weird fungus, and they really thought it was fascinating. They've been doing all sorts of studies on it, but commercial was getting frustrated. They're like, come on, we're a pharma company, not a research institute. You got to come up with something. And what are you going to use this for? And they're like, kidney transplantation. And apparently it was, as close as Swiss executives can get to rolling on the floor laughing, because it's like, There's no market for kidney transplantation. It just doesn't work. How are you ever going to make money on something that doesn't work? Well, the drug was cyclosporin. It made kidney transplants work. It became a billion-dollar drug because
Wow.
it created the market. And so, yeah, there's sometimes, to some degree, it's almost like a leap of faith. Like you try to get really, really good data. And I
Yeah.
have a story from my own past of kind of the other side where we had great data. And then after I left Infinity, the one drug cratered in a trial because, again, our ignorance should never lose track of the fact that while we understand enormous amounts about bio, our ignorance is huge. Like another way I always put that is I think about in terms of like the first year textbooks of different classes. So like my grandfather, my paternal grandfather was an electrical engineer, and he would have gotten his degree like early 1900s. His physics textbook, his first year physics textbook, probably didn't look very different from my first year, my 2 semester physics class that I took in the 80s, 90s. Because you think about it, it's mostly like motion, electronics. I mean, the
Laws of
actual
gravity
coursework,
haven't changed.
right? The actual coursework ended with Maxwell's equations, which is before Grandpa took, my grandfather took his class.
Yeah.
If you look at the intro chemistry class, then yeah, there was nothing in my intro that basically ended in like the subject material. I think basically ended in like the mid 20s. It's like with Lewis acids and so forth. My father, when he got his chemistry degree in the mid 40s, would have had all the same material. If you look at my OChem class, organic chemistry, very little of that Dad would not have had. There would have been like NMR, a couple of cage compounds, couple things. Most of that is really there. I am routinely finding things that I think should be in freshman bio textbooks that did not exist when I went to school in the 80s. I mean, the biology, we're still kind of working out those fundamentals. Like one I always point at, because I worked a lot on at Millennium, is one of the fundamental things we teach is homeostasis, the idea that in cells are always trying to kind of maintain a steady state, and so there's forces in either direction that cells manipulate, and you learn a lot in biology because it was worked out about how proteins are made, transcription, translation, ribosomes, and so forth. It was only when I was an undergraduate that we really started to tease apart how proteins are broken down. And that's a fascinating area. And again, there's this thing called the proteasome, and it looks like something out of science fiction or like a gigantic garbage disposal. It is like it's this long tubular like protein with a cap on it. Almost looks like a Hubble Space Telescope or something. But proteins go in and they don't come out. And it's critical. And you'd think, oh, it's just a garbage disposal. Get rid of bad proteins, get rid of proteins you're done with. But no, this is an important way to regulate a lot of cellular processes. It's really critical to the functioning of T cells in your immune system. This is how they get the little bits and pieces that are used to recognize whether cells should basically live or die. And we didn't, that work, which won a Nobel Prize, and Millennium developed one of the first drugs targeting it, or known to target it. It turned out an old drug worked in this pathway as well, thalidomide, a very notorious drug. But yeah, that we didn't start working that out till the first papers in that are around the time I started my undergrad career. So, and there's other things like, a lot of this epigenetics. I wasn't taught much of that as an undergrad because we didn't know that. Or even just emphasis. Like I was taught that alternate. So you have these introns, and sometimes you have choices there of like which exons you splice or what's considered an exon. We call that alternative splicing. And I was taught, oh yeah, that's a rare phenomenon. It's kind of this weird side thing. It's like, no, it's really, really important to so many things and so many genes have alternative splicing.
Yeah.
And alternative splicing gone wrong underlies a lot of genetic conditions. So it's this is really important. And it was kind of like soft, it was like, oh yeah, this is, you're not going to need to worry about this.
Yeah. So kind of bring this full circle with what we started talking about with sequencing. All of sequencing is critical to understanding how any of this works.
Yeah, it comes in a lot of ways. And one way to think about it, so at one level, for example, for rare genetic diseases, in many times, simply by sequencing patients, you can recognize the causative mutation.
Yeah.
And you can discover new causative mutations. And that leads to, in some cases, treatment options. And in some, unfortunately, relatively rare, but we're coming up with ways to treat patients just based on knowing that. It often uncovers new biology. You can also sequence the RNA, so what genes are expressed, and that tells you a lot more about the state of the cell. In like single cell technologies, you really do that on a cell by cell basis. So in a complex tissue, like say the bone marrow, which is just this all sorts of different cells, you can tell, okay, which cells are turning on which genes. And you can, of course, you can use that to discover how many different cell types are there, or you can do some kind of, partitioning. Spatial is doing that on like tissue slices. So you're doing single cell analysis and seeing which genes are turned on.
Yeah.
But one of the other very powerful things about sequencers is, and it was sort of hinted when I talked about molecular stenciling, is you can come up with clever ways to generate molecular patterns or just sequences that you then read out with a sequencer. And so a sequencer gets closer and closer to being a universal measuring machine if you can figure out how to convert what you're interested in into DNA. So one example is in like some single cell and spatial, we want to know what proteins are there. So what do? Well, you bind an antibody to the protein, and that's a very specific recognition. And if that antibody has a small synthetic DNA tag on it, you can now read those out with sequencers. Similarly, there's these technologies from like Olink and now Illumina protein prep, where you can take a blood sample, you expose it to Olink, their antibodies, in Illumina protein prep. There are these things called aptamers. But the same idea that things bind to protein, and they have little synthetic tags, and each tag's like a little barcode. And now you read those out, and I can read proteins out by sequencing DNA. And it gets crazier. Example I point to that's just not in biomedical, but George Church, who was my other advisor, had a preprint once on how to detect dark matter using DNA sequencers. I mean, this is, getting way out there, but it is. So in drug discovery, there's a lot of processes that you can turn into. Did I see a gene go up or down? Can I, like, another one that's out there is, suppose, the expression, what genes are being expressed at what level. Think of that as a fingerprint of a cell. And so we can fingerprint all sorts of normal cells and recognize them, that you show me this pattern and I say, oh, well, that's obviously a white blood cell, or that's obviously a neuron. Now imagine something like a cancer cell. In part, the cancer cell has a different profile than the cell it came from. So if this is a lung cancer cell, it looks different than the lung type of lung cell that was its progenitor.
Yeah.
Well, what if a drug could move the cancer cell closer to the correct state? So that's a way to search for a drug, is to go looking for drugs that move things or knock out genes or add genes and see which of those affects. Like, if I perturb the cell in some way, what moves a cancer cell more like a normal cell, or a normal cell more like a cancer cell? And getting back to the epigenetics theme, there is actually a set of drugs, and the first one was called IDHIFA, that alter. I mean, there are a number of drugs that alter epigenetic state, but this is an interesting example where a cancer mutation in a very central gene called isocitrate dehydrogenase causes a new small molecule, a new metabolite to form that's not normally seen in the body. And that turns out to be an inhibitor of an enzyme that's needed to do some epigenetic modification. I haven't gone through this in a while. I forget a few of the details, but the key bit is in these leukemias, what basically happens is a cell gets stuck in a juvenile state. It's supposed to progress on to another state. And because the epigenetics is wrong, it's stuck there. And they multiply and multiply, and they squeeze out everybody else. And you also are not getting the matured cells you should. So this is what causes, under normal circumstances, ultimately a fatal effect from leukemia. So then if you come in with a drug that interferes with this mutant isocitrate dehydrogenase, which is IDHIFA, and there's now follow-ons, then you block the production of this metabolite, you end up restoring the epigenetic state, and now these cells can escape. It's what we call a redifferentiating therapy. So the cells now go through their normal lifespan and die like they're supposed to, rather than being stuck. So, you compare it to, imagine if someone never, like there's movies like this of someone stuck being an obnoxious teenager forever, an eternally living obnoxious teenager. And so by changing the epigenetic state, you get them out of that, and they go on to lead a productive adult life and terminate, which is what cells are supposed to do.
So with every step of the drug discovery, drug development process, sequencing is like the eyes in which you observe.
It's an eye, and companies are, particularly like Element, really pursuing the strategy, but all of them are trying to get more and more into drug discovery. There's plenty of drug discovery processes that have no DNA sequencing.
Yeah.
But there's, the bull case for a lot of sequencing is we can replace a lot of with sequencing, or we can just get much broader eyes. And you can do these things like, with the protein profile I was talking about, you can just profile more proteins at a larger scale than was really practical by any other method. And
Yeah.
so,
So
everything gets bigger, and by bigger, humans are complicated. So the more we can look at, the more we can understand the big patterns and the small patterns and the noise and all that.
this is actually a great segue into my next question, which is how does it differ to sequence DNA versus RNA versus proteins? Is that every
Yeah.
layer you have different types of technologies that are used. So we can start
Yeah.
at the DNA
Sure.
layer. You have, short read sequencing, long read sequencing. You have different ways of doing it. Oxford Nanopore uses, nanopore
Yeah.
technology, which kind of my understanding is just it's like this little chip which sends different signals based on which base pair is going through the holes in these chips.
Yeah.
And that's nanopore tech. And you have, I don't know, I forget what the tech that Illumina uses. And I don't know enough about transcriptomics, RNA sequencing, and then you have proteinomics, which is protein sequencing.
Yeah.
And I know that you need to use mass spectroscopy
Yeah.
for protein sequencing usually. That's my understanding so far, but I feel like there's a lot I don't know in this landscape. And yeah, how does it all?
I mean, for better or worse, I could fill like a day. It's such a rich space. Some of the basics out there. So in DNA, basically, there's two major split in the technology. There's short read and long read.
Yeah.
And it does get down to a bunch of technical details of how you do it. But the key bit is short read is short. So like we're talking like 150, 200, under 1,000 bases generally. The advantage, so whereas long is like thousands of kilobases, the world record, it's really hard to do this, but for Oxford nanopores is about the size of the E. coli genome. It's at 4.2 million bases, which is really crazy. They differ in their error rates. They differ, but particularly for like some of this drug discovery, the short read, I can generate many, many more reads. And so for a lot of these things we've discussed, like protein profiling or RNA profiling, to some degree, they're a counting exercise. And the statistics depend on the number of counts you have. So more counts are better. And many, many, many more counts are much better than a lot, but not nearly as many. So
Yeah.
short reads really excel at that. Also, Illumina has been the market leader for a long time, so they're very entrenched. And so a lot of times it's, well, that sounds really cool, but not only is it they've spent the money on the Illumina, they've spent the money on the upstream processes like lab automation, the software automation downstream. There are different tools to analyze the data. Some of that's the different error profiles. Some of it's just if you've taken certain tricks with short reads, they won't work in long reads. Particularly like the error, because of the error, a lot of these tools were developed around the error pattern of short reads. If you want to get into the tech terms, things like De Bruyne graphs, some of those are really messed up by the error profile of, say, nanopore sequencing. So they will not work so well. But yeah, so you have these different techs, different tools. They're expensive. So like Nanopore, some of their instruments are only like a couple $1,000s. The biggest short read sequencers are like $1.5 million, and awesome in size. Like that Nanopore sequencer will literally fit in your shirt pocket. You hook it up to a laptop, you can take it anywhere.
Yeah.
It's been used on the International Space Station.
Yeah. The ion Q. No, not ion. What the mid ion.
Yeah, so the mid-ion can go everywhere. There's like an Illumina NovaSeq X is like the size of a deli fridge. It's a beast. I mean, there used to be the early PacBio instruments for optical stabilization literally had it was either ton or 2-ton slab of granite in them. That isn't anymore, but is like some fun, back history of those.
Yeah.
But so they all do different things. For RNA, most times what you do is you use an enzyme called reverse transcriptase to convert it to DNA, and then it's DNA sequencing. For Nanopore does have an option to sequence the RNA directly, and RNA also has epigenetic tags on it, which is very much the situation that I can't tell you how important most of those are because there weren't good ways to read them until recently. Nobody knows. Until you get the tool, you don't know whether it's worth, what is. It looks pretty fascinating. It looks like, again, there's all sorts of things.
Yeah.
And then, proteomics, if you really want to sequence proteins at scale, then you need a mass spectrometer. They're big instruments. They're expensive.
Yeah.
They require a lot of skill. there are a bunch of companies that are trying to develop new methods of sequencing. Quantum Sci is the only one that has a product,
Mm.
and there's it's definitely a really new field. It's fascinating. All of these, the science is crazy. So, like you said, Oxford nanopore, you're threading a single strand of DNA down through a pore and watching the electrical signal change as that DNA interacts with the pore. PacBio uses a quantum mechanical trick, so they can watch a single DNA polymerase extending a single molecule of DNA. But and it would be amazing if they do that on one, but they're doing it on like millions on a sensor chip. Yeah, I mean the Illumina technology you look into, there's all sorts of just crazy biochemical trickery,
Yeah.
and all of these. It's again, no matter how much you think you know them, you can always go and discover more details, more little just interesting little quirks of them that
Yeah.
can sometimes it's just trivial knowledge, and other times, yeah, if you're trying to develop, we try to develop a NGS, so sequencing based approach to SARS-CoV-2 diagnosis during the pandemic. It did not work, but it really, in part, got tripped up by some really, really fine details about the Illumina NovaSeq platform.
I see.
It's not
Before
Illumina's fault. It's just there are just little details that most of the time you do not need to worry about. We had to worry about them.
before we jump to the biotech market side of things, one last question. If we unlock massive proteomic sequencing, proteomics just
Yeah.
ubiquitous, what does the world look like differently?
Yeah. Well, one is, of course, you're going to need the data processing and I mean, that's one of the things. Like someone was just telling me, one of these spatial technologies, every time she runs it, she generates a terabyte of data. And
Right.
I know in some fields that's not trivial, but again, it's like we like large N in drug discovery, any kind of biology. So if it's 1 terabyte per slide, and you start saying, "Oh, let's automate this and set it up so we can run 1,000 a week," hard to predict.
Yeah. Yeah.
But it will change things because ultimately, the other thing about it is like we developed high throughput DNA sequencing, and that tells us a lot, but that's not what's really doing things in the cell. Then we developed the high throughput RNA sequencing, and for the most part, there's exceptions. RNA is not what's really happening in the cell. What we really want to know are proteins, and ultimately, where a protein is can sometimes matter as much as whether the protein's present or not. So, protein in the wrong spot, disease, or there's this protein called NF kappa B. Like, if it's in the cytoplasm, it's inactive. If it's in the nucleus, it's doing its work. Very important for cancer or inflammation. But again, if you just measure NF kappa B levels, that's not really telling you what you need to know. You need to know where it is. And then the other thing about proteomics is, again, there's the 20 amino acids most commonly put in proteins, but they can be modified post translational modification. And like one I worked on a lot at Millennium that of particular importance is there are proteins called kinases that add a phosphate group to 3 different, well, it's really 4, but mostly 3 amino acids. And that can radically change the shape of a protein, its
Yeah.
lifespan, its function. It can activate, can deactivate it, cause it to be destroyed. It's cause it to localize differently, causes it to bind different proteins, all those things. So yeah, proteomics is getting closer and closer to real biology. Like all of these are kind of like,
Hmm.
if like the Plato's allegory of the cave, like we're seeing shadows on the wall. Well, this is kind of like DNA is kind of like the shadows of the shadow of the shadow of the shadow kind of a thing. Like it's gone to or you're looking through very murky glass. Each one of those is getting closer to the actual biology. But yeah, so if we just had high throughput proteomics of bulk materials, it would change things. If you could do that single cell, even better. If you do that with spatial resolution, even better. It's always, we're always greedy that way. We keep wanting, and of course, it's a thing of scale. Like I was saying to people, I remember in the early, it was basically 15 years ago, when a single cell biology paper meant exactly that. People sequenced a single cell,
Yeah.
like the RNA, and published it on one cell.
Yeah.
And now, of course, people talk about doing these atlases of 1 million cells.
Which company are you most excited about in proteomics or sequencing?
One or two. Oh yeah, this is hard. I think the next gen proteomics is not clear that there's a winner yet. Quantum Sci is out there, but it's too low powered. There's very limited the kind of applications for it. There's a number of other companies out there, but they're all, again, it's really bleeding edge tech, and so they're all working other things. I mean, the fact Oxford Nanopore is just amazing. I always tell a story. So in George Church's lab, he is one of the two threads at Harvard. There are two groups at Harvard, unbeknownst to each other, until they got to Harvard's patent office, basically their technology office. We're working on the problem. But we had a really talented, really nice postdoc, Rich, was working on it. And he had he had his early setup inside a Faraday cage on an air table to isolate everything. And yet still, he could tell who was walking down a hallway, who he could not see. There was no window in that direction. He could tell by the pattern, like he was able to recognize each of our kind of cadence of footsteps on that there. And so like to go from that to this device that you can put in your pocket and literally have it sequencing when it's in your pocket and doesn't have one pore, it has 512 pores on it. It's just mind blowing. It took a lot of work to get there. Yeah. And of course, and if you look, there's a lot of technologies in this space that people have tried that ran into dead ends. Now, maybe if they've been funded better, or in some cases, it's also like a timing. Oxford Nanopore made, I know this from someone who told me, Oxford Nanopore made this big announcement in 2012 at a big genomics conference, AGBT, and that literally killed funding, like an attempt to secure funding for a competing idea using electron microscopy. So there's all this like chance in how we get to where they are. But so like in the proteomics field, it's hard to say. In the DNA field, I know people at all of them. I think they're all exciting technologies. I think also like Ultima is very exciting just because they keep dropping the price of sequences. People are like, oh, this is a race to the bottom. Well, as a biologist, I love these races to the bottom because every time someone there's a quantum drop in the price of sequencing, then the ideas that you previously thought were absolutely preposterous, people can do pilot studies. And the things that were pilot studies become semi-routine, and the things that were semi-routine become routine because the cost keeps going down. And again, like that first bacterial genome at Tiger was probably several $1 million, and at Ginkgo it's like a few tens of dollars to sequence a bacterial genome, and that just changes what you can do and how you perceive it. Like the whole idea in bacteria, for example, the whole idea of one bacterial genome is kind of nonsensical. Bacteria are just really, really plastic in their genomes. Similarly, even in the human space, like again with rare diseases, the fact that this was a huge change. Like Millennium did these studies in those days. If you wanted to work out a gene for a disease, you had to get a lot of families, build a big pedigree, and follow it through there. This is the only way that you'd be able to detect where in the genome that gene is. With rare diseases, you just find several people with that rare disease and sequence them, or ideally, like find a affected individual and their two parents and sequence it, because some of these diseases are de novo mutations usually. Neither parent has it and it's a dominant disorder. And so the only way is by sequencing the affected individual. And that's been enormously powerful. It worked out lots of things, but you can only do that because the price of sequencing went, the first human genome was like probably $100 million. I forget when Jim Watson's was done, it was I don't know, it was at least $500,000. I think it was more than that. I mean, for a while, it was like Steve Quake, someone in this field, did his, and it was estimated it was $100,000. Now that the genome, you can do a genome for a few $100. Like, it changes what you can do. So dropping the price is really exciting. Illumina has some cool tech coming out. Nanopore is always pushing things back. I love them all.
Okay. Well, do you have thoughts on long read versus short read? Because my understanding is that the price of long read is steadily going down over the years, and there's latent market demand for long read sequencing, and short read. It's just cheaper to do short read now. It's becoming more commercial.
So short read's always going to have an advantage in counting. It's also like there's a way of preserving medical samples called formalin fixed paraffin embedded. And getting the DNA back out of those samples, it's amazing. You store things at room temperature. It preserves the microscopic structure. You can literally have these things sitting on a desk for decades, and it's fine. Right. But when the DNA comes out, it's really beaten up, and it's really short. So long reads will never have an advantage there. Similar, if you've heard about liquid biopsies, the idea, if you go into blood, there's DNA floating around there, and that can tell you about the state of the body, particularly in cancer, but that's also how some of these prenatal tests work and so forth. That DNA is also very short. Most of it's like only a few hundred bases. It's like 150 base pairs long. It's basically one turn around a histone is the predominant one. So there, long reads will never have an advantage. I think for human genomes, I Illumina has this interesting new technology called Constellation, which basically tries to get long range information out of short reads. And there's some others out there, but it looks very intriguing. But I'll still think long reads are the right way to get long range information. And so, in like the rare disease space, I think long reads will predominate. But yeah, I think short reads will, they're just always going to have an advantage in the number of things they can get. So any kind of counting application, any of these like omics things, tags, like synthetic tags. There's also kind of a middle ground, which is the new Roche sequencer, which is very exciting.
That's a
Xelios.
the Roche, is that a PacBio sequencer?
No, it's so Roche a long time ago had a partnership with PacBio, and they dropped them. No, they've
Okay.
brought their own. It's a nanopore based device, but it doesn't really, other than it uses a nanopore, it doesn't have a lot of properties the same as Oxford nanopore because they have to synthesize. So this is just crazy. So the in the Oxford nanopore, the pore is roughly 10 bases deep. And so this is one of the things is like, whereas every other sequencing platform, I can sit down on a blackboard and like show you a signal, like you take a picture of signal and we could sit down and work those out. On nanopore, you get this complicated signal because there's like 10, most of the interactions in the center, but you're getting some from the rest. So like 10 bases, so what, 4 to the 10th, that's a very large space. You're not resolving that number of signal. So even if you got lucky, it's not like there's a simple fingerprinting. So it's just they have to have very nowadays it's machine learning to basically watch, as it's stepping through there, how that signal changes. You can then use that to resolve to a base. It's also complicated by the fact, and this is why they can read out methylation, is basically any molecular difference alters that signal. So when I said 4 to the 10th, I lied. It's a much worse. It's like 8 to the 10th or something because of modifications, damage, and all these other things. Or not terrible, but it's a fascinating problem, and Nanopore's done an amazing job of solving it. The first Nanopore data came out, I got thrilled because it was a 48 kilobase read, which was just unheard of in those days. But also the data barely matched. It was like 65% identity. They've gotten
Right.
it up into the high 90s now. It just
Right.
largely through better software. So the Roche sequencer makes a copy of your input molecules with these really weird nucleotide analogs called expandamers. And they basically look like you have a nucleotide. Imagine a nucleotide's like a VW bug. It has the Empire State Building parked on top of it. Except that well, not quite. It's not quite that scale, but it's like imagine like a big scissor lift on top of it, and then they do a chemistry and it splits it. And so the key bit is now after expansion, the pore is one expandamer wide. So they go back to Roche gets this simple signal of, and they very much like metronomically, it's like a soldiers in formation. One base goes in, they measure. Then they can ratchet the next base in and measure, ratchet the next base. Oxford Nanopore, it's a little noisier. You'd like to think it's that way, but it's a little bit noisier. So the Axelios one has this one, it's very fast, and they may have seen they set a new speed record for sequencing a human genome and delivering a clinical report. But also, they can generate a lot of data in what I call a MIDI read range. So like around a kilobase, like a kilobase to a kilobase and a half. And that can be interesting. But again, it's like, oh, we had short reads and we have long reads, and now we have MIDI reads. Things a little bit. Oh yeah, I think so. I think they're all going to have important impacts in biomedical research for the foreseeable future.
Yeah.
None of them are like this perfect, like Star Trek tricorder technology that's going to stomp everyone else out and you just wave it, kind of the general direction of what you want to know about and you get everything. So
Okay.
I think that
Yeah.
they're all cool. There's a bunch of startups out there trying to come up with even more technologies. I know of a few. Every now and then they pop up on my radar. They're all very, typically very, very stealthy.
Yeah.
So we'll, watch this space kind of thing. It'll never be boring.
Let's talk a bit about Ginkgo now. You've been at
Sure.
Ginkgo since 2019, I believe.
Yeah.
You've kind of seen the meteoric rise of Ginkgo when it went public in what, 21, and the subsequent just sort of lull in the biotech market.
Yeah.
I think it's just a fascinating story of how Ginkgo captured the imagination of so many people with the promise of synthetic biology and maybe overpromised what was possible in the short
Yeah.
term. And is now kind of slowly, consistently working to realize that promise. Could you help us understand what is the promise of synthetic biology and where does Ginkgo fit into all of it?
Yeah, so Ginkgo's vision, and many have had it. I mean, it came out of George Church's lab, and he's been thinking about this sort of thing for since before I knew him. I mean, there's some visionaries this, and actually, it kind of goes back to there's a famous essay by Richard Feynman that there's plenty of room at the bottom, which is
Yeah,
a lot of nanotechnology, but really, biology is nanotechnology. You can control atoms,
yeah,
but it's the idea that there's many things. There are many ways to get value out of being able to control biological processes or have biology make things. So instead of synthesizing useful products using complex chemical reactions that generate lots of nasty waste, for example, can we do those same things in with biological systems? And so, for example, when I was at Codon, we were working on something that, if you look at nylon, there are several classes of nylon, and they're basically the length of the carbon chain that is then connected together using amide bonds. And so the three carbons in a row back in, this would have been about 2008, 2009, or 2007, 2008. About half of the market was made by petrochemicals, and the other half was made by a process where E. coli made these monomers, the things that you then link together to make nylon. And so that's an interesting possibility. It's also things like even things we get from nature, but are hard to get. So imagine different, especially in flavors and fragrances, drugs. I mean, we found a way to use the inherent biology to make it, but the first patient ever treat it with penicillin, they knew they were going to run out of it, so they were prepared for this. They actually re-extracted it back out of the man's urine. If you've ever been on penicillin therapy, you will realize every time you hit the bathroom that the main way penicillin leaves your body isn't broken down, it goes out through the urine. The poor guy still died, but they got a really clear clinical response and then figured out, and then people figured out there's a long history there, which unfortunately there's not a good book for, to make it by the ton. But there's a lot of compounds out there in biology. They're really rare. And so if we can make them, and there's the simple, where's synthetic biology, where's molecular biology? It's not worth arguing. But like insulin, insulin, it used to be that it was pig insulin and you had to, make huge quantities of, access slaughter yards and so forth. And now it's made in E. coli and
yeah.
that,
That
and there's many others. We look at, I mean, it used to be a standard biochemical thing was if you wanted to extract some human protein, you went to a slaughterhouse and just hear about these things, just enormous quantities of offal, and then would run through them to get exquisitely tiny quantities of the final protein.
yeah,
And now what do? We clone it and produce it recombinantly in an
yeah,
appropriate system. And
yeah.
that's led to all these protein therapeutics
Mm.
and so forth. So you can also go in the other direction of, well, what if you have, bioremediation where you try to break down some of the mistakes we've made in the past, or bioconcentration. Like there's a lot of interest right now in rare earth materials and the geopolitical aspects of that, and how important they are for a modern economy, a modern defense space program. And so
You're saying we could
they're
synthesize rare earths
not
in
synthesize, extract. So like, how do
ex
you get them
okay,
out of, there are elements, so we can't make the element,
parts.
but when you have a pile of ore,
Yeah.
how do you extract it out? And in particular, suppose you have a pile of low grade ore, where you have the tailings from things that are lower, all extraction of like elements, it's like it depends on the market price and what's the quality of the ore you can get, like when it becomes economically infeasible. So might you be able to extract this, or especially what about recovering these rare earths back out of discarded apparatuses that we built with it?
How would you do that by engineering a specific enzyme or a protein
Yeah,
that
like engineering proteins to bind those rare earths.
got it?
I know at Codon I came up. I mean, we didn't pursue it, but then it was like rubidium and ruthenium, and the way those are mined up in Canada out of these, basically they're side products of mining nickel,
Yeah.
and you end up with these gigantic ponds of water contaminated with organic solvents that also have low concentrations of these valuable metals in them. So I
Mm.
don't know whether it's practical, but my idea was, okay, let's engineer an organism to, yeah, it would basically, one way or another, you bind those molecules to concentrate them. Use the, and again, a lot of times if you're fighting, like to move something from low concentration to high concentration, you need energy. Well, we can feed the cells things, and we can have the cells do all sorts of transporting against a gradient, like going, moving things uphill in a chemical sense.
Yeah.
So yeah,
That's
so
so interesting. I feel like there is renewed commercial interest in something like this, and I have no idea what the
I hope
state of
so.
the art. There is definitely is. I just don't know what the state of the art is today.
Yeah,
That is good
it's
homework
definitely
assignment.
something I just know exists. So yeah, so that was always like the promises, and then there's the even well you saw, and it kind of we rode and fell with the fake meats kind of concepts,
Yeah, yeah.
and then yeah, you could also, then there's these even more like out there but cool out there things like self-healing concrete, or things that you embed bacteria or fungi in something, and they in a material, and somehow they change the property in useful ways of it, or in like in self-healing, like they would basically go, the idea is they would go to a crack and remineralize it. If you look at our bones, the way, why are bones so strong and durable? It's because they're constantly being remodeled. So if you really want it like a structure that never fell apart, I mean, this is way science fiction-y, but you would simulate bone, and you just have
How
these cells in it constantly turning over the material, finding the defects and
how
rebuilding them.
how science fiction-y is it? Because I think
Oh,
when you first hear about synthetic biology, this is what gets people excited.
yeah.
Instead of using concrete, you're using self-healing bone-like structures. That's
There's
so cool. That is so cool.
in society to drive some of these things. I mean, it's often cheaper in society just to throw stuff out. But with especially making concrete is horribly uh, on the carbon budget. It's a
Yeah.
lot of carbon gets put in the atmosphere by conventional concrete, Portland cement production. Which
Yeah.
is another idea. Like, could basically have cells make Portland cement? So, and I don't know that space well enough, but I know that there's a lot of people who think about these things of how to
Yeah.
do it. And so, yeah, Ginkgo's concept was we would provide this central foundry
Yeah.
where you could make synthetic biology happen. So in the dream, and now we're turning that into, we will have an automated lab that
Hmm.
lets you run your experiments at scale, because a lot of times you do need high numbers, and kind of most automation, a lot of laboratory automation, I shouldn't say most, but a lot of laboratory automation is very much fit to purpose. So it's like having a custom integrated circuit for this task and another custom integrated circuit for that task. And maybe you can modify it, a bit, but they're still pretty much made to purpose. And we're trying to build an automated laboratory that is closer to sit back and, especially me who's a klutz in the lab. So sit back, think of the experiment you want to run, and then you just kind of feed materials in and data flows out. That would be the grandiose vision. We're making steps in that direction, where we have, we've done kind of these automated lab in a loop things where you generate 50,000 data points in a day or so, a day or two, and
Yeah.
feed that into a model and come back out. And I've been thinking about a blog post that, the sort of thing that you might do with that in the biomedical space, in this rare disease space,
Hmm.
to feed into, to generate very specific kinds of data for very specific models where we know that the existing machine learning models are not good enough.
Can you
And
give an example of that?
yeah, so I'll just go to this example. Hopefully I'll still write about this, but a lot of mutations that cause rare disease, one large class is that they affect splicing, and they damage splicing of introns and exons. And there's models out there. Splice AI is a very heavily used one that work a lot of the time, but there's a lot of the time they do not. Like Splice AI goes, that looks fine to me, and it's a devastating mutation. And so you could imagine from a software standpoint, well, these are not mutually exclusive, but if you're going to put your money on it, is it more that you need better models or you need more data? And I would almost certainly say we need more data. That the models probably didn't have nearly enough data and is overfit. So, I mean, I've toyed with a bunch of different ideas, but the idea would be, the general concept would be, can you have this automated laboratory run experiments to generate much larger data sets of given a genetic variation in the signals for splicing, how does that affect splicing? And so you'd run this, generate a lot of data, feed it back into a model, and then you maybe generate a new set of data and use that as your test set. for that initial trained model. I have to figure some of these out. One of the things I've just was drilled into me early on. I'm not a machine learning person, but you want to be really, really careful about your test and keeping your test and training sets.
Yeah, no, that makes sense. What is your role at Ginkgo today?
Yeah, so I'm in a product group, and the idea is so a lot of my time is spent reaching out to scientists, and then we organize these interviews. And if anyone wants an interview, just come reach out. And we basically ask, and our initial focus was in next-gen sequencing labs, but we're broadening that. We're interested in all areas: drug discovery, proteomics, flow cytometry, et cetera. But what's your experience been with automation? What are the areas where you haven't achieved as much as you'd like to? What are the things you haven't even tried to automate? And what drives things like purchasing decisions, which is purely commercial. But I think that the most interesting part, there's a lot of automation out there. Like when I first offered this position, I was like, wow, this is amazing. And then I started looking, it's like, oh my gosh, this is a crowded space. There's a lot of stuff out there.
Yeah.
But when you talk to people, there's gaps. So some of it's like software. Software is hard to use. People remark that they have to go to the manufacturer to get new protocols built, or they can't tune the protocols. One of the things interesting to me, I do not have much hands-on, as in nearly zero, experience with automation. I know next-gen sequencing is these robots. Like some people really want to, people want to be able to program them at a high level to say, at the highest level, like move the experiment, do this. But people also often have a real need to be able to control these things at exquisitely detailed levels, like move this piston this much,
Right.
kind of thing. So it's like, imagine, like, especially as a programmer, I always have tended to work at like high level. Let's find a bunch of really powerful Python libraries, tie things together. And this is like, well, we want to do that, but we want to be able to reach down to the not machine code, but the microcode in the processor. This is like, wow. It is. And there's good reasons to do that, because automation can do a lot of amazing things. You can make do things that are too complex for humans. You can have experiments run at times humans aren't. It's not practical. There was one time at Millennium, I was like, why don't we have a data point here? It's like, well, someone would have had to come in at 2 a.m. to collect that data.
Oh
like,
God, wow.
Yeah. You can get around that kind of thing.
So there's like the software piece of it that's a constraint. There's the granularity piece of it that's a constraint.
Granularity.
Does Ginkgo have its own proprietary software for running?
So what we have is, so like there's a lot of good instruments out there. And so what we do is we build hardware that ties them together. So like you can have like, things going from step A to step B to step C in a way that other companies, at least some of the solutions out there, don't. There's certainly similar ones. But the other is, it's software layers to try to make it easier to program these. And also things like, so if you have this complicated, I go first, like my samples need to go to this station, then to this station, then to this station. That's what it is. It's a track. I call it like, toy trains for biologists. But also like you can go and simulate that. And so and we're working on more and more sophistication of that simulator. So you can figure out problems, like figure out your mistake. I'm a klutzy programmer. Programming, it's just not as expensive to screw up usually as it is in the lab. So things like, oh, did you take the lid off your plate? Did you tell it to do that kind of a thing? Did you, or some kind of a cover? Did you overflow a well? Kind of mistake, kind of things that you should be able to figure a lot of that out in software before. And then also, we always go for best in class solutions. Like one thing you discover when you talk to people, and these lab robots are great, and there's some, but they're also like, and I noticed this before when I was at Warp Drive, that a lot of them are dumb robots in that kind of like old fashioned So like if you tell it to pick up a supply, a box of pipette tips, and that you forgot to put those tips on the deck, it'll just assume they were there. It'll go through the motions as though it was there. It will not go, oh, wait a minute, there isn't actually a box of tips there. So like we go for solutions that are like that. Look at the hardware, look at the ones that are the most precise, because you hear there's definitely, it's good. We're scientists, we're skeptical. And some of it's people who've been burned, where automation hardware has made mistakes. And it's one thing to make mistakes that burn money. It's really painful if the mistake uses up a sample that's irreplaceable, which is very common. biomedical research.
Yeah, no, that these are all really great points. Um, let's wrap up with a
Sure.
couple questions. I'm debating whether or not to ask the first one first. Who are some people in biotech that really excite you? When you talk to them, you go, "They're the future of the industry. I want to spend as much time with them as possible."
Oh boy.
I ask
Oh,
this in a selfish capacity because I also want to talk to them. So I'm just
yeah. No, that's a great one. I think. Well, so Andrew Sturgakis, I think is how he says his name, up at University of Washington's developed a lot of these like molecular stenciling kind of techniques. He calls it something else, and I apologize for forgetting. But I've never met him face to face. But he does a lot of cool stuff. Boy, yeah. I'm gonna lose track of names. There's a lot of people who've done amazing work on getting this final human genome and like solving these really, really difficult parts of the genome. Adam Philippe's one that his name just popped in my head that have done amazing stuff. I guess it's look at my blog. People I bring up there. Yeah, like who else? Boy, yeah, that's a that's a tricky one. So all the people I think are really cool, and names are escaping me right now. I apologize.
that's okay.
But yeah, I like anyone who's doing things on like kind of the bleeding edge of what can you do with sequencers, what can you do at scale. Yeah.
Okay, well, I'll take that. And the last question, what have been some of the most meaningful pieces of media, books, articles,
Yeah. So
that you would recommend to everyone?
sure. So I think everyone in biotech should read The $1 Billion Molecule. It's the
Right.
story of the early days of Vertex. He wrote a sequel, which isn't quite as good, but it's a fascinating look at both some history. It works in some history of medicinal chemistry, but also of their, a company trying to figure out what it's going to do. Personality, a certain amount of personalities pulling apart. Just interesting interesting story. His second one, so if that's the infancy, his second one is kind of like the preteen years. It's too bad there's not the one in the middle or the ones following, because Vertex is a fascinating company where they've had to basically pivot multiple times. And in fact, they had a successful drug that got destroyed by someone else's drug. And so they had to pivot then. And this final pivot has been the one I said would never work, and it's been enormously successful. So hats off to them. But it's a great book. It's also not a super thick read. Kind of a nice compliment that came out like a year ago. It's called For Blood or Money,
Hmm.
which is also about two different companies that were developing leukemia drugs and their story. And it's also like, this is definitely one to, especially people new to the space, need to read, because there's a little bit of kind of elbows flying in $1 Billion Molecule. This one, I mean, it starts out with a guy sobbing in his car because he's just been fired over the phone, just to give you some idea. I mean, it's not all that, but it is like, yeah, this is, there's a bunch of hurt feelings in here, and bruised egos, because people played hardball. And I'm not condoning it. Like, some of these people are like, I do not want to work for these people at all. Another book that was I read as an undergrad, sorry, and is quite good is The Coming Plague by Laurie Garrett.
Hmm.
And it's about like pandemics and disease detectives and so forth. And it was written during some HIV, and HIV was still kind of untreatable in that. And so it's interesting to see how well in general that's aged. I mean, we do have better molecular diagnostics. It's also, having gone through COVID, just kind of depressing how many of the bad patterns and mistakes that she documents there, and especially for HIV, were repeated with COVID. As a society, we still have a ways to go on pandemic preparation. That's another one. There's the ASEQ Discord, which is run by Nava
Yeah.
Whiteford, who runs a blog also called ASEQ. That's a good read.
That's how we met. Yeah.
Okay, all right. That's how. All right. Brian Kruger has a great blog. He used to have a paid one. He stopped doing that because he got a full-time job again. The Asimov Press guys write all sorts of fascinating historical stuff. I will confess, I do not read them enough. I love
very interesting, very wonderful.
I love their stuff. I went to a trivia night they had, and they, it was like, well, if I had read their stuff more carefully, I would have done a lot better. But they're great. I feel like there's there's a lot more. So Derek Lowe in the pipeline. Reading him was one of the major inspirations for starting my blog. He's much more consistent. He's always fascinating. He's very educated. He's a small molecule drug developer, but he has a very broad vision. Also, my good friend, Josh Gellikar of The Curious Wave Function. He's written in a lot of different areas. He's now writing a lot on AI and chemistry, but he also has really cool, no biology, but really cool stuff on the history of like theoretical physics, Oppenheimer, the U.S. nuclear program. Let's see. I'm trying to think of any other names I should drop. There's probably a bunch more, but those are
This,
all great ones.
this is a great start. No, I mean, start isn't this is going to take some time to work through. I'm really
Yeah,
excited.
I always warn people. Like, I'll have these. Like, I did a talk at Boston University, a career talk, and I told people, if feel free to reach out to me, connect to me on LinkedIn. It's all these students. I love interacting with students. And I said, if you talk to me and I give you all these like reading things, I'm not expecting you to read all of them right away or ever. They're just suggestions because
Yeah,
I can
yeah,
just
of course.
keep spitting them out.
Of course. No, this is great. Thank you so much. This was wonderful. I really
Thanks
appreciate you
for
taking the
inviting me.
time. Is there any last thoughts you have that you want to share? We can wrap it up here.
Probably should just wrap it up because otherwise I'll go. It's just, I mean, I guess it's just biology is fascinating. We're in a century of biology. This is becoming one of the dominant technologies.
Yeah.
I think and things like AI are really exciting, but there's always going to be need for experimental methods to feed the AI to check the results.
Yeah.
And yeah, and most importantly, we can really change human lives. That there was a great video shown at AGBT this year. A kid that was just his epilepsy was getting worse and worse. He was losing the ability to walk, he'd lost the ability to walk, lost the ability to talk. By genetic sequencing, I mean, this is one of these amazing stories we'd like everyone to experience. Not everyone does. By sequencing, Kingsmore, another really exciting group in the US, was able to identify the mutation, was able to come up with this customized drug called antisense oligo, and they were just starting treatment, and the boy was responding right away and improving. I think he'd regained the ability to walk but not talk yet. They
Wow.
were, and they had just started, and that's the sort of thing. And like I said, like Vertex, how they've changed cystic fibrosis patients' lives, or some of the cancer drug and cancer. There's still a long ways to go, but in some areas, like Gleevec, just radically changed lifespans for people with CML. Biology can make amazing differences in people's lives, and I would like to see that
Yeah,
continue.
well, that's a great way. That's a great note to end this on. Thank you so much, Keith. Um,
Thank you.
I hopefully will see you again in Boston at some point, but
Yeah.
until then, this was wonderful. I'll keep you updated on things.
All right. Great. Thanks.
Awesome. Thank you, Keith. Bye bye.
Bye.